స్టార్ట్ చేసి చెప్పండి సార్. ఓకే,
వచ్చేస్తుందా?
అని ఒకసారి చెక్ చేసి
Good morning, students. Today I'm going to explain you structural organization of proteins.
వస్తుందా? చెప్పండి, నేను ఒకసారి చూస్తాను.
Yeah, okay. Now here as a part of this amino acid chemistry, so today I'm going to explain you how the proteins has been undergo various levels of structural organization. So this is an important topics for you. It is an important topic even in the exam point of view. It is an essay question for you, and try to follow this carefully. So here the structure in the structural organization of protein already as we know that proteins made up of one or more polypeptide chains, and proteins with the single polypeptide chain is known as a monomer. Okay, proteins are like made up of one or more polypeptide chains. Proteins with the single polypeptide chain are called monomeric protein, and while the proteins with the one or more polypeptide chains we can call it as oligomeric proteins. Monomeric proteins are predominant in nature. Okay, proteins may contain if the single polypeptide chain, that we can call it as a monomer. If more than one polypeptide chain are there in the structure of a protein, that we can call it as oligomeric proteins. But generally most predominant proteins, most of the proteins are monomeric proteins. Now each polypeptide chain in a oligomeric protein we can call it as a subunit or a monomer. Okay, in a oligomeric protein each polypeptide chain we can call it as a subunit, or else you can call it as a monomer. Examples of oligomeric proteins is hemoglobin. Hemoglobin is, it contains made up of 4 polypeptide chains. Now here out of the 4 polypeptide chains, 2 are alpha chains and 2 are beta chains. So total 4, total 4 polypeptide chains are there in the structure of a hemoglobin. Hemoglobin is an example of oligomeric protein, whereas myoglobin is a monomeric protein as it contains single polypeptide chain, whereas hemoglobin is a oligomeric protein with the 4 polypeptide chains. And even lactate dehydrogenase is an enzyme which is also a oligomeric protein, like it has the 4 polypeptide chains. Now each protein has a unique 3-dimensional structure which is referred to as native conformation. Okay, whether it is a oligomeric protein or it is a monomeric protein, each protein has its own 3-dimensional structure which is referred to as native conformation. Okay, and in the native protein conformation, proteins do not exist as linear or the single dimension form. Instead they can they exist as the coiled structure or folded structure or the 3-dimensional conformation. So only such 3-dimensional conformations are biologically active. I think you are following. Okay, monomeric proteins means with the single polypeptide chain. More than one polypeptide chain in the structure of a protein is called oligomeric protein. But generally proteins here has a unique, each protein has its unique 3-dimensional structure. So here that is called native conformation. Okay, in the native conformation, how the protein exists, they will not exist in the linear form, they will exist in the biological fluid in the 3-dimensional in the form of a folded structure or the coiled structure. That folded or the coiled structure is known as 3-dimensional conformation. So such 3-dimensional conformations are biologically active. So when the proteins are formed, means when the proteins are synthesized on the ribosomes, how they will be synthesized in the linear form. Okay, proteins on ribosomes, they may be synthesized as linear form. In order to attain 3-dimensional structure, means for the formation of the active 3D conformation, proteins has to undergo various levels of structural organization. Okay, originally proteins on ribosome synthesized as the linear sequence, but in order to attain three-dimensional structure, the protein has to undergo various levels of structural organization. Okay, so that structural organizations are, each protein is organized into four levels of organization, that is primary, secondary, tertiary and quaternary structure. Okay, see here, in order to attain, once it is being synthesized, in order to attain the three-dimensional structure, protein has to undergo various levels of structural or four levels of organization, that is primary, secondary, tertiary and quaternary. Okay, now here this secondary primary second this secondary, tertiary and quaternary structures are called higher structures, and monomeric proteins, they show only means monomeric proteins, they show only up to tertiary structure. Okay, if the protein which is synthesized on the ribosome, if it is a monomeric protein, in order to attain three-dimensional structure, it will undergo primary, secondary and tertiary structure. But only the oligomeric proteins, you know that oligomeric proteins means which has more than one polypeptide chain, exhibit quaternary structure. For example, you take myoglobin. I already mentioned myoglobin is a monomeric protein having single polypeptide chain, shows only the organization up to tertiary structure. For example, you take hemoglobin. Hemoglobin is a, like it's a hemoglobin is a oligomeric protein. In order to attain three-dimensional structure, means proteins which are having more than one polypeptide chain, they need to undergo, along with these three, they will undergo even the quaternary next organization, that is quaternary structure. Okay, that's it about the introduction about the levels of structural organization. I think you understood that why the protein has to undergo structural organization, means once it is synthesized in the linear form, so in order to attain three-dimensional structure, to become fully active, it needs to undergo various levels of organization: primary, secondary, tertiary and quaternary. Monomeric proteins undergo the levels of organization up to tertiary structure. Whether it is a like oligomeric protein, they will undergo all four levels of structural organization. Now here, what is the hierarchical nature of the primary structure? What it refers, what is primary, secondary, tertiary and quaternary? Means primary structure which refers to the amino number and sequence of the amino acid. Primary structure refers to the amino number and sequence of the amino acid in a protein. And secondary structure, folding, twisting of the primary structure to form secondary structure. The most common patterns of the secondary structure are alpha helix and beta sheeted. And the next one is the tertiary structure. Tertiary structure is the three-dimensional structure formed by the assembly of the secondary structure. So that means further folding of the secondary structure to form tertiary structure. So tertiary structure is a three-dimensional 3D structure of a protein which has been formed by the assembly of the secondary structure. And the next one is the quaternary structure. We know that it is the structure which is formed by the proteins which are having more than one polypeptide chain. So these are the hierarchical nature of the protein structure. Primary structure denotes the number and sequence of the amino acids. Secondary structure, the folding of, twisting of the primary structure to form secondary structure, alpha helix and beta sheeted. These are the regular patterns, common patterns of the secondary structure. And the tertiary structure, it is a three-dimensional structure formed by the assembly of the secondary structure. And the next one is the quaternary structure. Oligomeric proteins are having more than one polypeptide chain undergo the next level of organization, that is quaternary structure. So this is the quickly to understand primary structure. As I mentioned, these are the sequence, number and sequence of the amino acids, and the folding of the primary structure to form, the twisting of the primary structure to form secondary structure. The secondary structure patterns are alpha helix and beta sheeted. Alpha helix is the spiral one and beta sheet is the extended form of the primary, and then further folding of this secondary structure, okay, to form the tertiary structure. And the proteins which are having more than one polypeptide chain can show the quaternary structure. This is the quickly understand about the different structural organizations of the proteins. Okay, even you can see here, these are the proteins have the different structure levels of organization. Primary sequence of the amino acids. So like this, see here the folding of this, twisting of this to form secondary structure, and the twisting, further folding of this secondary to form the tertiary structure. Now here, and the tertiary structure on further folding in case of oligomeric protein to form assembly of this to form the quaternary structure. Now here, coming to the even this picture also showing you how the proteins is organized into various levels of organization. Okay, primary, secondary, and three-dimensional structure is the tertiary structure, and this is the complex of the protein molecule, that is quaternary structure. Now coming to how this secondary tertiary structure, how the twisting and folding has been stabilized means, so there are various bonds are there, the few different bonds are there which are responsible to stabilize the protein structure. Okay, now let us discuss about the bonds involved or the bonds responsible for protein structure. So the bonds which are responsible for maintaining the protein structure is peptide bond, ionic bond, disulfide bond, hydrogen bond, hydrophobic interactions, and even the disulfide bonds. Please note down this one here. The bonds, the folding of the secondary, tertiary and quaternary structure or the primary structure, how the amino acids has been maintained, how this primary second has been maintained stabilized means by various bonds. The bonds which are responsible for the structure of the protein are the peptide bond, ionic bond, disulfide, hydrogen bonds, hydrophobic interactions, and even the disulfide bonds. Now here all this, so the bonds are of two types. So the bonds which can stabilize the protein structure has been divided into, based on their strength, divided into covalent bonds and the non-covalent bonds. Okay, covalent bonds are the strong bonds. Examples of the covalent bonds which stabilizes the protein structure are peptide bond and disulfide bond. And the non-covalent bonds are generally weak, but collectively, as they are present in combination in various proteins, they give strength and stability to the protein structure. Okay, so the covalent bonds are strong enough to maintain the structure. Examples are peptide bonds and disulfide bonds. Now here the non-covalent bonds are generally individually they are weak, but most of the times they are present in combinations, so they give strength, stability to the protein structure. Examples of non-covalent bonds are hydrogen bonds, hydrophobic bonds, ionic or electrostatic bonds, van der Waals forces. So these are the various bonds which involved in the protein structure, which stabilizes the structural organization of protein. Now here coming to the peptide bond, we already know that how the peptide bond has been formed. The alpha carboxyl group, okay, alpha carboxyl group of one amino acid as react with the condenses with the alpha amino group of the next, okay, another amino acid to form a peptide bond. Okay, peptide bond is formed by the reaction or the condensation between the alpha carboxyl group of one amino acid to the alpha amino group of the another amino acid by using one water molecule to form a dipeptide. Okay, here this is a dipeptide, amino acid 1, amino acid 2 joined by peptide bond to form a dipeptide. Okay, this is the peptide bond, the one, this color, okay, this color is seed of the peptide bond is formed between the carboxyl group of one amino acid to the amino group of the another amino acid. And make a note, the carboxyl and amino groups are has been joined to the alpha carbon. Okay, and we know that so proteins are made by the polymerization of the amino acids through peptide bonds. So peptide bond joins the two successive amino acids. So proteins are the polymers of amino acids. So how they will be polymerization of amino acid occur means through this peptide bonds. So peptide bond is the then, so based on the number of peptide based on the number of amino acids joined by peptide bond, already we know result in the formation of dipeptide if 2 amino acids are joined by one peptide bond, tripeptide if 3 amino acids are joined by 2 peptide bonds, like that, so on. Tetrapeptide, 4 amino acids joined by 3 peptide bonds. Oligopeptide means more than a few amino acids are joined together by amino acids, result in the formation of oligopeptides. And more than 10 to 15, more than 10 and up to 50, they can result in the formation of polypeptides. And we know that if more than 50 amino acids are joined, result in the formation of a protein. So that is about the peptide bonds. So what you have to understand here, peptide bonds are the bonds, they are the covalent bonds, join the alpha carboxyl group of one amino acid to the alpha carboxyl group of the next alpha carboxyl group of one amino acid to the alpha amino group of the another amino acid to form various peptides based on the number of amino acids. Now coming to the next one is the disulfide bonds. So disulfide bonds are also a covalent bonds, strong bonds. So how they will join, how these disulfide bonds are formed between the sulfohydryl groups. SH means sulfohydryl groups of the side chain of the cysteine in the same or the different peptides. Okay, see here, if two cysteine, if suppose you take one single polypeptide chain, so these disulfide bonds are formed between the sulfohydryl groups of cysteine within the same polypeptide chain, or the sulfohydryl groups of cysteine of one protein, one polypeptide chain has been linked to the sulfohydryl groups of cysteine of the second polypeptide chain. Okay, so that means there are two types of disulfide bonds are there, interdisulfide bonds and intradisulfide bonds. First, what you have to understand, what is a disulfide bond? So what is it? It is the bond which is formed between the sulfohydryl groups of the side chains of the cysteine. Okay, means this is the bond which is formed between the sulfohydryl groups of the two cysteine residues. That two cysteine residues, if they lie in the same polypeptide chain, to form intradisulfide bonds. Okay, if the sulfohydryl groups of the cysteine lies in the two different polypeptide chains, to form interdisulfide bonds. Okay, so like that, even the disulfide bonds also give stability to the protein structure. They are the strong bonds. So these disulfide bonds are most common in like connective tissue protein keratin and the extracellular enzyme, that is ribonuclease. Okay, disulfide bonds are most common where? Here in the connective tissue protein, that is keratin, and extracellular enzyme, that is ribonuclease. And function of these disulfide bonds is they stabilize the they protect the structure of protein from the denaturation and also confers additional stability. So that's it about the two covalent bonds, peptide bonds and disulfide bonds. Now coming to the next one is hydrogen non-covalent. Now let us discuss about the non-covalent bonds. And I already mentioned non-covalent bonds are non-covalent bonds are individually weak. Name the non-covalent bonds: hydrogen bonds, electro van der Waals forces, ionic bonds, and hydrophobic interactions. All these are the examples of non-covalent bonds, and these bonds are individually weak, but combined of various combined of various non-covalent bonds contribute stability to the proteins. So one of the non-covalent bond is hydrogen bond. How the hydrogen bond is formed? By sharing of the hydrogen between the nitrogen and carbonyl oxygen of different peptide bonds. Okay, see here, I repeat here, how the hydrogen bond has been formed means, this is the hydrogen bond formed between the amino group NH group and CO carboxyl groups of peptide bond by sharing the single hydrogen. Okay, so that means hydrogen bonds are formed between the CO and NH groups of a peptide bond by sharing a single hydrogen. Okay, now how this bond is formed means, the electrical attraction between the hydrogen atom. Okay, the electrical attraction between the hydrogen atom in a polar bond in one molecule and oxygen or nitrogen in a polar bond in another molecule or within the same molecule. Okay, how the hydrogen bond means, there is a electrical attraction between the hydrogen atom of a polar bond in one molecule and oxygen or nitrogen in a polar bond of another molecule or within the same molecule. Okay, so if the bonds are sufficiently separated from one another, it forms a hydrogen bond. So that means what you have to understand, hydrogen bonds, okay, in a protein molecule, the hydrogen bonds are formed by the sharing of the hydrogen atom. Okay, in a protein molecule, the hydrogen bonds are formed by the sharing of the hydrogen atom between the nitrogen and carbonyl oxygen of different peptide bonds. Okay, now there is a hydrogen in each residue. There is a hydrogen donor atom and hydrogen acceptor atom. So the atom to which hydrogen is bonded is called hydrogen donor atom. The atom to which hydrogen is shared, that one is called hydrogen acceptor atom. The strength of the hydrogen, actually hydrogen bonds are weak, okay, but as they occur in the large numbers in protein structure, they give stability to the protein structure. Okay, hydrogen bonds are generally, they are weak, but as they occur in the large numbers, they give strength and stability to the protein structure. And you know that hydrogen bonds are formed between the sharing a single hydrogen between the nitrogen and carbonyl oxygen of a peptide bond. And the strength of a hydrogen bond that depends upon the distance between the donor and the acceptor. Okay, the strength, okay. Okay, the strength of the hydrogen bonds depends on the distance between the donor and the acceptor. Okay, and now here the groups in polar side chains of the amino acids, they can form hydrogen bonding with the surrounding water molecules, which will increase their solubility. Okay, the side groups of different functional groups which are present in the polar side chains of the amino acids, they may interact, they can form hydrogen bonding with the surrounding water molecule, and thereby enhances their water solubility. Okay, enhances the solubility of the proteins. Okay, now here look at this picture. I already mentioned this is the individual residue amino acids. Now here the blue ones are the hydrogen bond donors. Hydrogen bond donor means the atom to which the hydrogen atom, the atom to which the hydrogen has been added. And hydrogen bond acceptor means the atom with which the hydrogen is shared. Okay, so the red one is the atom to which the hydrogen is shared, and the blue arrows represent the atom to which the hydrogen atom is bonded. So that is designated as blue ones are hydrogen bond donors and the red ones are hydrogen bond acceptors. The strength of a hydrogen bond depends upon the distance between the donor. The strength of the hydrogen bond depends upon the distance between the hydrogen donor and the hydrogen acceptor ions. Okay, and usually in the secondary structure, usually in the structure, in the protein structure, the hydrogen bonds can be represented by the dotted lines. Okay, in the protein structure, these hydrogen bonds are represented by dotted lines. And the next non-covalent bond is electrostatic bonds, ionic bonds, or the salt bridges. All are same only, electrostatic, or else you can call it as ionic bonds or the salt bridges. So these are the how they have been formed means these are the attractive forces between the two oppositely charging groups. Okay, electrostatic bonds are formed between the this is the formed between the two oppositely charges. For example, they occur between the oppositely charged groups such as carboxyl group of acidic amino acids and amino groups of the basic amino acids. Okay, so these electrostatic bonds are formed between the two oppositely charged groups, means carboxyl group of acidic and amino group of the basic amino acids, or they may be formed between the amino and carboxy terminal groups of the peptides. So like this, salt bridges, ionic bonds or electrostatic bonds, they have been formed between the oppositely charged groups. Okay, between the acidic and basic amino acids, or else between the amino group and carboxyl group of the peptides. Now next one, other non-covalent bonds involved in the protein structure are hydrophobic bonds. So these are the bonds, these are formed by the interaction between the polar hydrophobic side chains by eliminating the water molecule. Okay, so that means these are the interactions between the polar hydrophobic side chains by losing one water molecule. The property of the function of this hydrophobic bonds is they serve to hold the lipophilic side chains together. Okay, now here you have to make a note here. In the hydrophobic bonds, the hydrophobic interactions are not due to the attraction between the non-polar groups. Okay, here actually what are these hydrophobic bonds? These are the interaction between the non-polar hydrophobic side chains. But here you have to make a note that this is not the attraction. Okay, this is not the attraction between the non-polar groups, but due to the properties of the water molecules surrounding the medium, which push them together and resulting in the hydrophobic interactions. I repeat here, hydrophobic bonds. Okay, these bonds are not formed due to the attraction between the non-polar groups. So but how they have been formed? This is due to the water molecule which has been surrounding them, which push them together and forms the hydrophobic bonds. The function of hydrophobic bonds is which helps to serve to hold the lipophilic side chains together. And the next one is the other forces, bonds, non-covalent bonds are van der Waals forces. So these are the mainly attractive forces operating between all atoms due to their oscillating dipoles. So that means these are extremely weak. Van der Waals forces are extremely weak and they act extremely short distance between both polar and non-polar side chains of the amino acid residues. Okay, so individually these van der Waals forces are extremely weak and they act on extremely short distance between the polar and the non-polar side chain of the amino acid residues. Okay, that's it about the bonds which are responsible for maintaining the structural organization. What are the bonds? Peptide bond and the disulfide bonds. Disulfide bonds are of two types, interdisulfide and intradisulfide bonds formed between the sulfohydryl groups of the cysteine, and the other bonds are the hydrogen bonds. Hydrogen bonds are one line answer. They are found between the sharing the single hydrogen between the carbonyl and oxygen. Okay, nitrogen and the carbonyl oxygen of the peptide bond. Okay, and here the strength of the bond depends upon the distance between the donor and the acceptor. You know that donor is the atom to which hydrogen atom is bonded. Acceptor is the atom to which the hydrogen atom is shared, and hydrogen bonds are individually weak, but as they present in more number in the protein structure gives strength and stability. Now here the other bonds are electrostatic ionic bonds formed between the oppositely charged residues. Hydrophobic bonds are the interactions between the non-polar hydrophobic side chain, and van der Waals forces are the weak. They are formed between the short distances between the polar and non-polar side chains. Now coming to each structural organization in detail. Okay, so here till now what we have to what did you understand? So in order to attain the native in the native protein conformation, protein exists in three-dimensional structure. So to attain the three-dimensional structure, protein has to undergo the further folding process. Okay, originally it is synthesized, protein is synthesized on the ribosomes as the linear straight line. So in order to attain the three-dimensional, it has to undergo folding process. So in order to attain the three-dimensional structure, the protein will undergo various levels of structural organization: primary, secondary, tertiary and quaternary. And till now we have discussed what are the bonds which stabilizes or which are involved in maintaining these various levels of structural organization. Now coming to the first one is the primary structure of a protein. So primary structure is the sequence. Okay, what it refers primary structure, it denotes the sequence of the amino acids in a protein. So that means primary structure which indicates the number and sequence of the amino acids in a protein. The higher levels of organizations are decided by the primary structure. Even the biological activity of the primary of a protein, okay, the biological function of a protein also depends upon its primary structure. Primary structure, what it refers? It refers to the number and sequence of the amino acid in the protein. And Each polypeptide chain has, we know that it has a unique amino acid sequence, and a number of unique amino acid sequence that will be decided by the genes. Each protein has its own amino acid sequence, so that will depends upon that is decided by the genes. So the primary structure is maintained mainly by the covalent bond, that is peptide linkages. So primary structure refers to the linear sequence of the amino acids. So the linear sequence of the amino acid, how it is maintained, how it is stabilized, means by the peptide linkage, means peptide bonds. Now here, then each protein has its own amino acid sequence. For example, you take one tripeptide. Here the three amino acids are there. They have been arranged in the order glycine, alanine and valine. Okay, and then this is another tripeptide. Here the same amino acids are present, but their order is, they have been arranged in the order like glycine, valine and alanine. Okay, so both these tripeptides shown here are the same amino acids are present, but their amino acid sequence is altered. So that means what you have to understand, each protein has a unique amino acid sequence, even though they contain same amino acids, but each protein has its own amino acid sequence, and that will be decided by the genes. Now here, next one is here the primary the sequence of the amino acids in the protein is called the primary structure, and the primary structure is decided by the genes. If there is any mutations in the genes responsible, okay, for the synthesis of the proteins, that would result in the formation of abnormal proteins, and there is an impairment of the normal function is there. Okay, see here the primary structure is important here because which determines the physical and chemical properties of a protein, right? And primary structure which has the which refers to the sequence of the amino acids, that is decided by the genes. If there is any genetic diseases is there, genetic disorders are there, means if there is any mutations are there, that would result in the abnormal amino acid sequence. So abnormal amino acid sequence, that will code for the abnormal proteins which will impair the normal functioning of the protein. Okay, now here this primary structure is stabilized by primary structure is stabilized by peptide bond. Again, you know that peptide bond here is formed between the two amino acids. Here the amino acid is valine and the second amino acid is alanine, and there is a peptide bond is formed between the alpha carboxyl group of valine to the alpha amino group of the alanine to form valyl alanine. Okay, this is the dipeptide valine and alanine. They have been linked by through peptide bond to form a dipeptide known as valyl alanine. Okay, now here if you see this one, this for this one, and here this is the one amino acid, this is the second amino acid, and the water molecule is removed from here and to form a peptide bond. Now if you see this peptide, now the resulting dipeptide, what did you understand? Always in the dipeptides or in the peptides or the proteins, one side there is a free carboxyl group and on another side there is a free amino group. So that means here the peptide bonds join the alpha carboxyl group of one amino acid to alpha amino group of the another amino acid. First it forms a dipeptide. The amino acid chain length has been increased by the successive addition of amino acid to these free ends. Okay, each peptide has the free ends. One side there is always free carboxyl group that is not involved in the linkage, and another side there is always the free amino group is there. If another amino acid is there, that will be added to this free end. Okay, that is this amino group. This amino group of this will be joined to the carboxyl group of the second amino acid. Okay, like that. So always the peptide bonds are formed between the these amino acids are each polypeptide. Okay, each polypeptide has the free ends. Okay, that free ends are one side there is free carboxyl group and another side there is a free one side there is the free amino group and another side there is a free carboxyl group. So where the free amino group is there, that terminal is called N terminal. and the amino acids has been successively added to this carboxyl group to increase the length of the amino acid. So if you imagine the protein structure, always there will be a free amino group on the left side of the protein, and always there will be free carboxyl group on the right side of the peptide. The side where the free amino group is there, that is called N-terminal. Another side where the free carboxyl group is there, that is called C-terminal. Okay, I repeat here, amino acids, the proteins are the polypeptide chains made up of sequence of amino acids, right? How the amino acids has been joined in the primary structure by the peptide bond, and you know that how the peptide bond has been formed. Carboxyl group of the one alpha carboxyl group of the one amino acid is joined to the alpha amino group of the second amino acid. So now here, if there is another peptide bond, if there is another amino acid is added to this one, again the alpha carboxyl group of this may be joined to the alpha amino group of the next successive amino acid, like that. So on, amino acids has been joined to the peptide, to the polypeptides, through these peptide bonds. So if you see the structure of any polypeptide, one end obviously there is free amino group that is not involved in the peptide linkage. Another side obviously there is a free carboxyl group that is also not involved in the peptide. So always, so the one where the free amino group is present, that is called N-terminal. The other side where the free carboxyl group is present, that is called C-terminal. Okay, see here, that means when you name the peptide bonds, so always peptide chain, if you see the structure of any peptide chain, there are two free ends are there. One with the free amino acid, that end you can call it as N-terminal. One with the carboxyl end, that terminal you can call it as C-terminal. Okay, N-terminal amino acid is written on the left hand side and C-terminal amino acid is written on the right hand side. Except these N-terminal and C-terminal, all other alpha amino and alpha carboxyl groups are involved in the peptide bond formation. I think you are following in a peptide bond formation, except the N-terminal, okay, and C-terminal. Other all the remaining alpha carboxy and alpha amino groups are involved in the peptide linkage. Only there will be free amino group on the one side and free carboxyl group on the other side, and the rest of the remaining all the alpha carboxy and alpha amino groups are involved in the peptide bond formation. To the side where the free amino group is there, that side we can call it as N-terminal. To the other side where the free carboxyl alpha carboxyl group is free, that is not involved in the peptide linkage, that you can call it as C-terminal. Amino acid sequence is always read from N-terminal to C-terminal. Even proteins are also biosynthesized from the N-terminal to C-terminal. Please make a note here. The peptide bonds has the free N-terminal and C-terminal. Amino acids always read from which direction? N-terminal to C-terminal. And even proteins are also synthesized from N-terminal to C-terminal. Now here, how do you name this one? Glutathione. Glutathione is a tripeptide made up of three amino acids, glutamate, cysteine and glycine. Glutathione is a tripeptide made up of three amino acids, glutamate, cysteine and glycine. Generally, the amino acids which participate in the peptide bond, we do represent with the three-letter abbreviation. Okay, that is glutamate. No need to write glutamate. The abbreviation for glutamate is, we do represent in the amino acids with the three abbreviation. GLU represents the glutamate, CYS cysteine and GLY is the glycine. But when you name the peptides, when the peptide name for this glutathione is glutamyl, cysteine, glycine. So that means YL, the suffix YL is added to the name of the amino acid except the C-terminal. I repeat here, glutathione. It's a tripeptide. Tripeptide made up of three amino acids. Name the amino acids which participate involved in the synthesis of the glutathione or glutamate, cysteine and glycine. When you represent the name in the, when you, the peptide name for this is the glutamate, at the end suffix yl is added when you name the peptides. So glutamyl, cystinyl, but yl suffix is not added to the amino acid which is there at the C terminal. So hence the glutathione, the peptide name for the glutathione is glutamyl cystinyl glycine. Now here, okay, if you see this one, so always this is the N terminal and this is the C terminal. Amino acid 1, amino acid 2 joined through peptide bond that would leave the amino acid free on left side and carboxyl group free on the right side. So this terminal is called C terminal and this terminal is called N terminal. Always amino acids are read from which direction? N terminal to C terminal. Even proteins have been synthesized in the direction N terminal to C terminal. Okay. Now here, these peptide bonds, there is a characteristic. This is the even this picture also showing that amino terminal and the C terminal. Okay, carboxy terminal. Now here, this is the free amino terminal and this is the this is the N terminal, that is amino terminal, and this is a C terminal and where there is a free alpha carboxyl group. Okay, now here then naming the peptide bonds I have already explained you. So by convention, the free amino acids of the peptide chain N terminal is written on the left side and the free carboxyl group, that is C terminal, to the right side. Now here, what are these? There is a special characteristic features of there for their peptide chain. The peptide bond has the partially double bonds character. Okay, please note down. Till now we have discussed how the peptide bond has been joined, has occurred between the two amino acids. So there is characteristic feature of this peptide bonds. So that is, peptide bond is a partially double bond character and it is rigid planar. Okay, see here the peptide bond is the partially double bond character. It is like rigid planar, and other properties, other characteristic features are peptide bonds is a polar covalent linkage and the polar groups are involved in hydrogen bonding. Please note down the characteristic of a peptide bond. Peptide bond is planar. I'll explain you why it is planar. And the peptide bond has partially double bond character, and peptide bond is rigid, means there is no freedom of the rotation. Means freedom of the rotation is restricted, so it is rigid. And the peptide bond is a polar covalent linkage and the polar groups are involved in the hydrogen bonding, and all the peptide bonds in the protein exist in the trans form. So these are the characteristic features of this one peptide bond. Okay, it's a partially double bond. Always it exists in the trans nature. As it is a partially double bond, there is no freedom of the rotation, and peptide bonds are planar, rigid, and there see here why peptide bond is partially double bond in nature means here, if it is a single bond, the distance is 1.49. If it is a double bond, the distance is 1.27. But here the distance between the CO and NH of a peptide bond is only 1.32 angstroms. So that is the midway between the single bond and double bond. Hence the peptide bond is a partially double bond. Okay, why it is a partially double bond? It is not a single, it is not a double bond, because if it is a single bond, the length between them should be more than 1.49. If it is a double bond, the length is 1.27, but the length in the peptide bond, the length between the CO and NH is 1.32. So that is the midway between the single and double bond, which gives the partial double bond character to the peptide. So as it is the as the distance is less in between partially double bond, which restrict the rotation, okay, which prevents the rotation of the bonds. Okay, the side chains are free to rotate on either side of the peptide bonds. So here, as there is the double bond, the means okay, sorry, we'll explain you one by one. Then the side chains, the means there is a freedom as it is a rigid, because of the partial double bond there is no freedom of the rotation, hence the structure is rigid. So why the structure is planar? Okay, why this the structure is why the peptide bond is planar means, look at this picture here. It is a you see it's a peptide. Okay, it's in the tetrapeptide. What will happen because of the peptide bond formation, all the alpha carboxyl group and alpha carboxyl, alpha carboxyl and alpha amino groups comes into the same plane. Okay, because of the peptide bond, what will happen, the all the atoms lie in the same plane. So that means the alpha carbon atom and the CO groups of the first amino acid and the amino group and the alpha carbon atom of the second amino acid all will lie in the same, because of the peptide bonds all the atoms of the different amino acids they may lie in the same line. So that's what these peptide bonds are planar. And the next one is peptide bond is partially double bond in character. Why? Because the partial double bond character of the peptide bond is that depends upon the length between the carbon. Okay, length between the CO and NH group I already mentioned. Because the length is in between, it attains the partially double bond character. So as it attains the partially double bond character, it prevents the rotation around the axis of the bond, as it exists as the, because of the partial double bond character, the rotation around that prevents the rotation around the axis of the bond. So no freedom to rotate because of the partial double bond character, that limits the movement of the peptide backbone. Okay, as the peptide bond is the partially double bond, as there is no freedom to rotate, that restrict, okay, that limits the movement of the peptide backbone, but making the bond essentially planar. Hence the whole structure is considered as planar. Peptide bond is polar covalent linkage which carries no net charge. Okay, the other point is peptide bond is the polar covalent linkage which carries no net electric charge. Okay, there is no net charge at that. Peptide bond has no it's a neutral, no net charge. So the uncharged nature of this protein helps in the folding of the protein. Okay, the uncharged nature of the protein allows a peptide backbone to be compactly packed in globular structure. I repeat here, when the protein is folded, it has to compactly folded into the globular protein, because the peptide bonds are the bonds which joins the two amino acids, as they are the uncharged, which help in the easily compact folding of the proteins in case of the globular proteins. Okay, now here, so these are the characteristic features of this one peptide bond. Peptide bonds are planar in nature. Because of the peptide bond, all the atoms are in a single plane, which gives that one. And because of the partial double bond character depends upon the length between the CO and the NH groups. So as the length is in between the single and double bond, which gives the partial double bond character to the peptide bond. So that prevents the restriction movement of the backbone of the peptide bond at the axis. Because of that one, means that there is no freedom of the rotation. Now here, and the other point you have to remember is, as there is the peptide bonds are uncharged, which help in the compact folding of the protein in the globular proteins. Now here there is a bonds and angles which are adjacent to peptide linkage. So that means, so the bond length, unlike the rigid peptide bond which does not allow the rotation, the bond adjacent to the alpha carbon and the carbonyl and the amino group and the alpha carbon, which are flexible and they are pure single bonds. Okay, in the pure peptide bonds which happens in between the amino acids are what? They are partially double bond structure. But the angles which are adjacent, especially to the alpha carbon and the especially to the around the alpha carbon and the carbonyl group and the amino group and the alpha group, enabling the protein to structure. That means what you have to understand. So there is peptide bonds are, there is no freedom to rotate peptide bonds. But the bonds which lie between the alpha carbon and the carbonyl groups are the amino group and the alpha carbon. Okay, the bonds which occur between the alpha carbon and the carbonyl group and the amino group and the alpha carbon are flexible. They are pure single bonds. Okay, so the peptide backbone has freedom of rotation. Okay, the backbone has the freedom of rotation around these bonds of each amino acid, which enabling the folding of the protein. So please, I repeat these points. Actually, peptide bonds has no freedom of the rotation, but there is a flexibility is there for the bonds, especially in between the alpha carbon and the carbonyl group and the amino group and the alpha carbon. Because of the flexibility, this will enable the protein to fold when it is organized into secondary structure. Okay, the rotation around these bonds are known as torsion bonds. They have been measured between -180 to 180. Okay, so especially the bonds, the peptide backbone has the freedom of the rotation around which bonds? Alpha carbon and the carbonyl group and the amino group and the alpha group. In between the bonds are freedom of rotation, which enabling the protein to fold. Okay, so the rotation around these bonds, except these, the rest of the bonds, peptide bonds, are there is no freedom to rotate, but especially these bonds, because they are flexible, they can rotate. So the rotation around these bonds are known as torsion bonds or dihedral bonds, and they have been measured between -180 to +180. So there are these angles are called, proposed by the scientist Ramachandran angles. Okay, so how these bonds are determined, means these are proposed by the scientist, that is called the Ramachandran, who was awarded Nobel Prize for that. So the Ramachandran angles are phi angle and psi angle. Okay, what is pi angle? Pi angle is the angle of rotation around the bond between the nitrogen and the alpha carbon. Okay, please note down, there are Ramachandran angles are two. Okay, that is psi angle and phi angle. Phi angle means this is the angle of the rotation where around the bond between the nitrogen and the alpha carbon, and psi angle is the angle of rotation around the bond between the alpha carbon and the carbonyl carbon. Okay, look at this one. He is the Ramachandran who did a pioneered work on the structural aspect of the proteins. For that he has awarded Nobel Prize. Now here the two Ramachandran angles, the torsion angles. What do you mean by torsion angles? The rotation around the, specifically I mentioned these bonds are known as torsion angles. There are two torsion angles are there. So that is, this is psi angle and this is phi angle. Okay, you look at this picture. Phi angle is the this is the rotation, angle of rotation around the alpha carbon and the nitrogen. Okay, and what is psi angle? The angle of rotation between the alpha carbon and the carbonyl carbon. Okay, so at these areas there is freedom to rotate. As the primary structure, at these bonds freedom to rotate, they can fold in the protein structure. So this phi and psi, phi angle is the angle of rotation between the alpha carbon and nitrogen. Psi angle is the angle of rotation between alpha carbon and the carbonyl carbon. Okay, these two are the Ramachandran angle, which are known as torsion angles or dihedral angles. So torsion angles means what? The rotation around these bonds, the rotation of these around these bonds are known as torsion angles. Now what is the importance of these angles? Means the Ramachandran angles, phi angle and psi angle, they determine the path of the polypeptide chain. Please make a note, these torsion angles which determine the path length of the polypeptide chain, and they also determine the extent of twist and turns of a polypeptide chain when it is folding into higher levels of structural organization. Please make a note here. The importance of this Ramachandran angle, usually peptide bond which connects the amino acid is there is no freedom of the rotation around the axis, but some areas between the alpha carbon and nitrogen and alpha carbon and the carbonyl nitrogen there is freedom of the rotation, that is known as torsion angles. Two torsion angles are there, pi and psi. Phi occur the rotation between the alpha carbon and nitrogen. Psi means the rotation around the alpha carbon and the carbonyl oxygen, and these angles will determine the path length of the polypeptide chain, and they also determine the extent of twisting or the turns in a polypeptide chain when the polypeptide chain is folding into next to higher levels of structural organization. Okay, and the primary we know that primary structure sequence of the amino acid that will determine the biological activity of a protein. Example of this primary structure is well explained in the insulin. Insulin is a hormone which is a polypeptide. It has two peptide chains are there, A chain and B chain. A chain is made up of 21 amino acids and B chain is made up of 30 amino acids. So total 51 amino acids are there in the structure of the hormone insulin. It's a it's a dimer. Insulin is a dimer made up of two polypeptide chains, and A chain has 21 and B chain has 30 amino acids are there. Now here in the A chain there is inter and intra disulfide chains are there. So inter 7th cysteine residue which is there in the 7th place of A chain is linked to the 7th cysteine of the B chain. Okay, how this A chain and B chain has been linked means there is a disulfide bridges are there between the cysteine which is there in the 7th position, 7th cysteine that will be attached to the 7th cysteine of the B chain, and similarly there is 20th amino acid in the A chain is cysteine and 20th amino acid in the B chain is cysteine. They both the cysteine sulfidyl groups of these cysteine has been linked by interchain disulfide bridges. Okay, that means A chain has 21, B chain has 30. A chain and B chain has been linked to each other through interchain disulfide bridges. How these interchain disulfide joins 7th cysteine of the A chain and 7th cysteine of the B chain, and 20th cysteine of the A chain has been joined to 19th cysteine of the B chain, and sometimes intra-disulfide bridges are formed between the same chain. Within the A chain only there is the 6th amino acid, that is cysteine. Okay, that has been joined to the cysteine which lies at the 11th position to this intra-disulfide bridges. Okay, like that insulin structure, like it's a dimer made up of A chain, B chain. A and B has been linked through intra-disulfide chain bridges happens between the cysteine residues of A chain and B chain, and within the same molecule, within the same polypeptide chain also intra-disulfide bridges are formed between the cysteine of the 6th position with the 11th one. Now here originally this is the structure of this insulin, and originally this insulin is synthesized in the beta cells of the pancreas as pre-pro hormone. Okay, when beta cells from the beta cells of the pancreas, the insulin is synthesized as the inactive form pre-pro hormone which has 110 amino acids. Okay, from that 120 amino acids the 24 amino acids has been removed under the post-translational modification to form pro-insulin. Okay, pre-pro insulin has 110 amino acid, that is the inactive form. From that 24 amino acid residues has been removed to form the pro-insulin which contains 86 amino acids. From the 86 amino acids, a peptide which is having the 33 amino acids from the center has been removed to leave the structure 53 amino acids, that is proinsulin. Okay, from that proinsulin 53, the next two amino acids has been removed by the carboxy peptidase to form insulin. Okay, now here this originally insulin is synthesized as pre-pro hormone. Pre-pro insulin has 110 amino acids. From that 110 amino acids, if you remove 24, means the 20 by the removal of 24 amino acids, it will be converted to pro hormone. Okay, pro hormone contains 86 amino acids. This pro hormone, by the enzyme convertase, it removes the 33 amino acid C peptide to form proinsulin. Okay, that proinsulin, by the removal of two more amino acids, by means that removal has been catalyzed by the enzyme carboxypeptidase to form the fully functional active insulin, that is 51 amino acids. So pre-pro hormone 110 amino acid, 2 by the removal of successive amino acids 24 amino acids, pre-pro insulin is converted to proinsulin. From the proinsulin, a C peptide having the 3 peptide bonds has been removed to form proinsulin, which contains 53 amino acids. From that 53 amino acids, 2 amino acids has been removed by the enzyme carboxypeptidase to become fully functional active insulin. So active insulin has how many amino acids? 51 amino acids are there, distributed in two chains, A chain and B chain. A chain is having 21 amino acids and B chain is having 30 amino acids, joined by inter and intra disulfide bridges are there. Okay, so like that the primary structure is explained by this insulin structure. Now the primary structure, you know that the number and sequence of the amino acids which will determine the biological activity, and the higher levels of organizations are depend on this one. If there is any mutation in the single amino acid, that will affect the biological effect of the function. For example, hemoglobin, example is hemoglobin A, that is a normal hemoglobin, sixth amino acid in the beta chain glutamic acid. Okay, the structure HbA is one of the normal hemoglobin. If you see the structure of hemoglobin A, at the sixth position glutamic acid is there. If there is any mutation happens, glutamate, if it is replaced by valine, that would result in the formation of HbS. So that is the abnormal hemoglobin that causes the sickle shaped RBCs, and they are easily ruptured. Lysis causes anemia. That type of anemia is called sickle cell anemia. Okay, that means you understood the importance of the amino acid sequence. If one amino acid is displaced by the another amino acid, that would but that would alter the biological activity. In the structure of hemoglobin A, at the sixth position, what should be there? Glutamic acid is there. By mutation, if glutamic acid is replaced by valine at the sixth position, instead of HbA, what is going to form? HbS is going to form. That alter the structure of the protein, so that attains, that RBC attains a sickle shape, that is easily fragile and easily ruptured because of the increased hemolysis of this HbS. There is which causes anemia known as sickle cell anemia. Okay, that's it about the primary structure of the protein. And the next coming class we'll discuss about the secondary structure, tertiary and quaternary structure, etc. Thank you. అయిపోయింది.